At this year's ASCO 2018, we had an update to the ALEX phase 3 study. Now, the ALEX study is a phase 3 study in advanced ALK-positive non-small cell lung cancer comparing the first-generation drug, crizotinib, with a next-generation drug, alectinib. And last year, we saw that it was clearly a positive study. The primary endpoint is investigator-assessed PFS. The hazard ratio is 0.47, but the median wasn't reached. The Independent Radiology Review Committee had reached a median, which was about 25 months. But what we've got this year with more than 20 months follow-up across the arms is now that primary endpoint, the investigator-assessed PFS, now has a median. And it's an amazing number. It's 34.8 months. And the hazard ratio has also dropped down to 0.43. Now, a lot of people are going, well, why is there such a big jump? And there are two reasons. So one is the primary endpoint is investigator-assessed PFS, and we know that investigators are always a little bit more generous than an IRC. The IRC was only done as a secondary endpoint and only done at the primary analysis, and we're not going to get that going forward. So we're always going to have this perhaps slightly rose-tinted interpretation of the PFS, but that's the data we have. The other reason is the median is occurring in a very flat part of the curve. And so a few patients one way or another can shift that median by a lot. And to be honest, although it's an easy number to remember, it's actually the hazard ratio, which is probably the best reflection of people when you have really long control of the disease. So the hazard ratio is 0.43. That's a 57% reduction in the risk of progression or death with this drug. To some extent, the updated results for ALEX are just a victory lap for the drug, but it's more than that. When you think about the sequential thought experiment of what happens if you went on crizotinib, progressed, and then went on alectinib, you would expect about 11 months with crizotinib, maybe about 8 or 9 months with alectinib given post-crizotinib. And when you're getting a median that is, somewhere between 25 and 36 months. I mean, really much, much better than you expect with sequential. You say you're changing something. You're changing the natural history of the disease. The other way of thinking about it is you are losing something if you do it sequentially that you cannot catch up with. So it really solidifies these next generation drugs, specifically alectinib, as what you should start a patient on. In terms of other developments for ALK, well, there's a number of things going on. So first of all, whilst people who are newly diagnosed, we're expecting them to probably start on alectinib, there's still a bunch of people on crizotinib. And when they progress on crizotinib, they have a range of different licensed drugs. I think we're starting to see mature data in terms of the fact that these next generation drugs post-crizotinib are not all exactly the same. It's astonishing how robust the median PFS is for a given drug, even between trials in that same setting. So, for example, ceritinib is 5 to 6 months. Alectinib, post-crizotinib is 8 to 9 months. Ensatinib is 9 months, they only have one trial. Lorlatinib, we saw an update, its median progression-free survival post-crizotinib is somewhere between 11 and 13 months. And brigatinib is the standout winner. It's between 16 and 17 months, and no one can explain why. It's clearly not just about the activity against ALK kinase domain mutations, because if you ranked it in vitro, you'd probably put lorlatinib at the top of the list. And what I think is these drugs have got other non-ALK activity, the secret sauce, which is allowing some of these drugs to have something more than what you can predict from the in vitro data. I think what happens is eventually, as you get better and better drugs at suppressing the ALK kinase domain, what you get is second drivers. And I think that some of the differentiating effects between some of these drugs is some of them can suppress some of the second drivers and some of them can't. So for example, crizotinib is also a MET inhibitor. And ironically, MET amplification has actually been recognized as a mechanism resistance to alectinib. So it's not that, crizotinib is done and we never need it. It will have little niche indications depending on what your mechanism of resistance is. Alectinib is also a RET inhibitor. Brigatinib has certainly some rather interesting effects. We don't entirely know what it is. Maybe EGFR related, may even be immune related. Speaking of immune, one of the other observations at this year's ASCO was a series of studies saying, what if you combined a PD-1 or PD-L1 inhibitor with an ALK tyrosine kinase inhibitor? Well, the take-home message is a terrible idea. None of the data suggests that the activity you see is anything other than just from the tyrosine kinase inhibitor alone. These were all done in usually populations where you'd expect the TKI to be working anyway, and mostly what you're adding is toxicity. There have been some fascinating retrospective studies proving the value of these kind of, really cheap, simple studies where people went back and said, look, when these immunotherapy drugs came out, I treated everybody. Well, how did it do depending on the driver oncogene you had? Internationally, the ALK positive response rate to immunotherapy is 0%. And it has been 0% in every trial where it's looked at it. And there's, if you had to choose a biomarker for who not to give monotherapy with a PD-1 or PD-1 agent to, it's ALK. In addition to the molecularly targeted subgroups where there are FDA-approved treatments, so that's ALK, ROS1, EGFR, and BRAF, there's a whole bunch of other molecular subtypes of non-small cell lung cancer in the wings, which are waiting to get a molecularly specific license for drugs. And I think these will also start to be carved out as things that you'd want to know up front rather than just giving everybody chemoimmunotherapy. And maybe the groups to look out for, I think maybe the next area is MET exon 14 skip mutations. These occur in about 4% of lung cancer. Crizotinib, which is also a MET inhibitor, got FDA breakthrough status approval recently, and that has about a 40% response rate. There's a Merck KGaA drug called tepotinib, sounds like teapot, tepotinib, which has also got about a 40% response rate. That has breakthrough status in Japan, and obviously they'll be looking for coming into other countries. At this meeting, we also saw information on rare EGFR and HER2 mutations called exon 20 insertions. That's about 10% of EGFR mutations. HER2 insertions is maybe about 2% of lung cancer overall. There had been some early data with a repurposed second-generation EGFR inhibitor called pazotinib, which was getting about a 60% response rate and a lot of toxicity. Here we saw a supposedly more rationally designed drug called TAC-32788, which believes that it can inhibit the drug with less effect on the EGFR wild type in the skin and the gut. It has about a 40% response rate. It's hard to say that's really different because the numbers are so small, and also not all exon 20 insertions may be equally sensitive, so you have to check that they're balanced between the arms. But that's also getting a fair amount of skin and gut toxicity. So I think it proves that these are actionable. I think we have to wait and see as these data sets mature in terms of whether these drugs are equivalent or different. And then RET gene rearrangements. There are two drugs at AACR this year. We saw Blue 667. At ASCO, we saw Loxo 292. And these are showing about a 60% response rate, including in that previously hard-to-treat KIF5B RET rearrangements. And then finally, the little sister to MET exon 14 skip mutations is high-level MET gene amplification. And crizotinib also works in that, but it's really high-level MET amplification. You have to look at the MET to centromere of chromosome 7 ratio. The latest data is saying it has to be 4 or greater, and we've got about a 40% response rate. But before you get too excited, that's about 0.5% of lung cancer. Now, if it was your mum or dad, you'd like to know it was there, but it's not anything a drug company is going to get that excited about developing as a big market.